Good day, this is Professor Will, CBMD PhD. Today is Thursday, April 30th, 2020. It is 3:31:57 seconds a.m. Eastern Standard Time. And this is a continuation of COVID-19 corona research. So let's get started here. New research highlights blood clot dangers of COVID-19. Date April 23rd, 2020. Source Radiological Society of North America. Summary. A special report published today in the Journal of Radiology outlines prevention, diagnosis, and treatment of complications stemming from blood clots in patients with COVID-19. The journal also published 2 research letters and a case study on this topic. Clinicians worldwide face this new severe infectious lung disease with no proven therapies. Based on recent reports that demonstrated a strong association between elevated D-dimer levels and poor prognosis, concerns have arisen about thrombotic complications in patients with COVID-19. The National Institute for Public Health of the Netherlands asked a group of radiology and vascular medicine experts to provide guidance for the imaging workup and treatment of these important complications. Their report summarizes evidence for thromboembolic disease and potential diagnostic and preventive actions that can be taken. From the analysis of all available current medical laboratory and imaging data on COVID-19, it became clear that symptoms and diagnostic tests could not be explained by impaired pulmonary ventilation alone. Recent observations suggest that respiratory failure in COVID-19 is not driven by the development of the acute respiratory distress syndrome alone, but that microvascular thrombotic processes may play a role. This may have important consequences for the diagnostic and therapeutic management of these patients. There is a strong association between D-dimer levels, disease progression, and chest CT features suggesting venous thrombosis. In addition, various studies in patients with COVID-19 have shown a very strong association between increased D-dimer levels and severe disease, poor prognosis. The report authors stress that careful attention needs to be paid to the initial diagnosis and treatment of the prothrombotic and thrombotic state that can occur in a substantial percentage of COVID-19 patients. Imaging and pathological investigations confirm the COVID-19 syndrome is a thromboinflammatory process that initially affects lung perfusion, but consecutively affects all organs of the body. This highly thrombotic syndrome leads to macrothrombosis and embolism. Therefore, strict thrombosis prophylaxis, close laboratory and appropriate imaging monitoring with early anticoagulant therapy in case of suspected venous thromboembolism are indicated. Recommendations for diagnostic and therapeutic management, which vary based on patient symptoms and risk profiles, include prophylactic dose heparin, chest CT, CT pulmonary angiography, and routine D-dimer testing. Findings have also emerged linking COVID-19 more specifically with pulmonary embolism. Researchers published today in Radiology reported that 106 pulmonary CT angiograms performed for COVID-19 patients over a 1-month period in a tertiary care center in France. 32 patients, 30% had acute pulmonary embolus, PE. This rate of PE is much higher than usually encountered in critically ill patients without COVID-19 infection. 1.3% or emergency department patients is 3 to 10%. In the study, a D-dimer threshold of 2,660 UGL detected all patients with PE on chest CT. Researchers pointed to a high proportion, 23% of COVID-19 patients with contrast CT had PE. PE was diagnosed in a mean of 12 days from symptom onset. Patients with PE were more likely require care in the critical care unit and to require mechanical ventilation. Researchers report and describe multiple areas of pulmonary and arterial thrombosis in an 84-year-old man with COVID-19. COVID-19 is more than a lung infection. It affects the vasculature of the lungs and other organs and has a high thrombosis risk with acute life-threatening events that require adequate treatment with anticoagulants based on laboratory monitoring with appropriate imaging tests as required. A new biosensor for the COVID-19 virus. Detection in the environment. Date April 21st, 2020. Source Swiss Federal Laboratories for Material Science and Technology, EMPA. Summary. Researchers have developed a novel sensor for detecting the new coronavirus. In future, it could be used to measure the concentration of the virus in the environment, For example, in places where there are many people or in hospital ventilation systems. Jing Wang and his team at EMPA and ETH Zurich usually work on measuring, analyzing, and reducing airborne pollutants such as aerosols and artificially produced nanoparticles. However, the challenge the whole world is currently facing is also changing the goals and strategies in the research laboratories. The new focus, a sensor that can quickly and reliably detect SARS-CoV-2, the new coronavirus. But the idea is not quite so far removed from the group's previous research work. Even before the COVID-19 began to spread, first in China and then around the world, Wang and his colleagues were researching sensors that could detect bacteria and viruses in the air. As early as January, the idea of using the basis to further develop the sensor in such a way that it could reliably identify a specific virus was born. The sensor will not necessarily replace the established laboratory test, but could be used as an alternative method for clinical diagnosis, and more prominently to measure the virus concentration in the air in real time, for example in busy places like train stations or hospitals. Fast and reliable tests for the new coronavirus are urgently needed to bring the pandemic under control as soon as possible. Most laboratories use a molecular method called reverse transcription polymerase chain reaction, or RT-PCR for short, to detect viruses and respiratory infections. This is well established and can detect even tiny amounts of viruses, but at the same time it can be time-consuming and prone to error. An optical sensor for RNA samples. Jing Wang and his team have developed an alternative test method in the form of an optical biosensor. The sensor combines two different effects to detect the virus safely and reliably, an optical and thermal one. The sensor is based on tiny structures of gold, so-called gold nanoislands, on a glass substrate. Artificially produced DNA receptors that match specific RNA sequences of the SARS-CoV-2 are grafted onto the nanoisland. The coronavirus is a so-called RNA virus. Its genome does not consist of DNA double-strand, as in living organisms, but of a single RNA strand. The receptors on the sensor are therefore the complementary sequences to the virus, unique RNA sequences, which can reliably identify the virus. The technology the researchers use for detection is called LSPR, short for localized surface plasmon resonance. This is an optical phenomenon that occurs in metallic nanostructures. When excited, they modulate the incident light in a specific wavelength range and create a plasmonic near field around the nanostructure. When molecules bind to the surface, the local refractive index within the excited plasmonic near field changes. An optical sensor located on the back of the sensor can be used to measure this change and thus determine whether the sample contained the RNA strands in question. Heat increases reliability. However, it is important that only those RNA strands that match exactly the DNA receptor on the sensor are captured. This is where a second effect comes into play on the sensor, the plasmonic photothermal PPT effect. If the same nanostructure on the sensor is excited with a laser of a certain wavelength, it produces localized heat. And how does that help reliability? As already mentioned, the genome of the virus consists of only a single strand of RNA. If this strand finds its complementary counterpart, the two combine to form a double strand, a process called hybridization. The counterpart. When a double strand splits into single strands, it's called melting or denaturation. This happens at a certain temperature, the melting temperature. However, if the ambient temperature is much lower than the melting temperature, strands that are not complementary to each other can also connect. This could lead to false test results. If the ambient temperature is only slightly lower than the melting temperature, only complementary strands can join. And this is exactly the result of the increased ambient temperature, which is caused by the PPT effect. To determine how reliably the new sensor detects the current COVID-19 virus, the researchers tested it with a very closely related virus, SARS-CoV. This is the virus that broke out in 2003 and triggered the SARS pandemic. The two viruses, SARS-CoV and SARS-CoV-2, differ only slightly in their RNA. And validation was successful. Tests showed that the sensor can clearly distinguish between the two very similar RNA sequences of two viruses, explains Jing Wang, and the results are ready in a matter of minutes. At the moment, however, the sensor is not yet ready to measure the coronavirus concentration in the air. For example, the Zurich's mainly railway station. A number of developmental steps are still needed to do this. For example, a system that draws in the air. concentrates the aerosols in it and releases the RNA from the viruses. This still needs developmental work, says Wong and researchers. But once the sensor is ready, the principle could be applied to other viruses and help to detect and stop epidemics at an early stage. Good day, this is Professor Will, CBMD PhD on infectious disease and microbiology. Well, I hope you enjoyed listening and have a great day.